The epigenome in a social context. Epigenetics in humans might not be very well understood, but the discourse about its social implications have been discussed. Many look to use it as a vector of transgenerational trauma, and there have been several studies that have showcased this possibility, although clear causality has not been established. Epigenetic transmission has been studied in the context of famines. The most famous study is of the Dutch winter famine. It is the perfect environment to study epigenetics in, as the population went from eating a relatively robust diet to a reduced diet and then back to a regular diet in a short span of time. It was found that being exposed during gestation caused significant epigenetic impacts. When comparing children who were prenatally exposed with their directly exposed elder siblings, significant differences in methylation were found. A similar study was done in the Överkalix parish in northern Sweden. They experienced a famine in the late 1800s, which led to positive and negative transgenerational effects. Through this study, it was found that there may be a link between starvation and the paternal line. It was even found that boys who had a greater access to food passed down more health issues that shortened lifespan than those who did not enjoy a similar access to food. In another study detailing the Great Chinese Famine, 1959 to 1961, with the Sui-Hua cohort, differences in exposed and unexposed populations in the methylation of genes that regulated autophagy were discovered. In autophagy, cells recycle unneeded and damaged cellular components by breaking them down into basic components to use them for necessary processes and energy production. Autophagy is needed to maintain homeostasis, but autophagy declines as one ages, increasing cellular stress and increasing mortality and the incidence of age-related disease. In fact, it was found in centenarians that autophagy-related mechanisms were maintained, which allowed for healthy aging. These famous studies into the transgenerational epigenetic impact of starvation has led people to ask if traumatic events like war or violent oppression have echoes in future generations. A study was done on Holocaust survivors and their offspring. It was found that there were some intergenerational effects of Holocaust exposure. The study was done on the methylation of the FKBPA gene. FKBPA regulates glucocorticoid receptors' sensitivity to glucocorticoid molecules like cortisol, which are essential in regulating metabolism, inflammation, and stress response. Variations in this gene have been linked to disorders like PTSD. Differences in methylation were observed across parent, survivor generation, and the offspring generation. Parent exposure even predicted methylation at some sites. A more recent study done on 3 generations of Syrian refugees also found epigenetic changes across 3 generations. There was the group where the F1 group, grandmother, had direct exposure to violence, and the F2 generation was in the womb, prenatal exposure, and the F3 generation had germline exposure. The second group was an uninfected F1 generation and an F2 generation that was directly exposed. The F3 generation was prenatally exposed, and their elder siblings were directly exposed. The third group was a control group, unexposed to violence. Prenatally exposed children had an older epigenetic age, biological age, and also in this group, exposure to violences caused difference in methylation across 35 sites. 32 of these sites were similar across all types of exposure. Despite these studies looking at epigenetics through a social context, specifically as a vector of trauma, it might be putting the cart before the horse. Epigenetic inheritance in humans is inherently more complex due to the fact that parental behaviors could also have an impact on the outcome of offspring. It is difficult to isolate epigenetics as a sole cause for a person's health problems. There are also significant downsides to the epigenetic model. Some believe that this will further promote biomedical reductionism, simplifying human processes, molecularizing environments and social contexts. Instead of creating an egalitarian policy, it could put all the blame on parents for social situations that they cannot control. For example, in Australia, Indigenous peoples are optimistic about the possible positives of using epigenetics to bridge a historical past with current biological consequences. However, it could also promote child removal policies using the justification of a bad environment, echoing earlier child removal policies. It is important to consider this trauma in the frames of the historical context. Removing the history of socioeconomic oppression belittles the circumstances of oppressed people and reframes them as inherent. When studying trauma, it could be helpful to study the effects in one community to ensure that the research is grounded in the context of the community. Epigenetics proved that the gene was dynamic, not set in stone, but to further employ it in a social context, like we have with genetics to a certain degree, the science and experiments done need to provide a definitive answer to epigenetic mechanisms and inheritance.